My name is David Anasatsiu, and I'm currently at San Jose State University, but starting in September, I'm going to be at Santa Clara University. And I'm going to talk to you a little bit about improving open modification spectral library search through exact filtering. Now, a little bit about myself first. I am not traditionally from computational genomics. I know very little about the biology. And the resource that was mentioned earlier today, I'm definitely going to look that up. I think that's really awesome. My background is computer science. So I do a lot of work in data mining and machine learning and high performance computing. But some time ago, I learned about some of the problems that you guys are dealing with and the types of interesting data that you're working with. So I became interested, and I started coming to CGSI 3 years ago, and eventually got involved with one of these interesting problems that you guys are working on. So to give you a very quick overview of some of the challenges that I'm tackling in general, I work on traffic analysis with people from NVIDIA and a bunch of other universities. We put together a challenge, a yearly challenge, which had a workshop at CVPR, the major conference in vision this year and last year as well. So the idea there is to solve problems, traffic related problems by analyzing videos. We have all these cameras all over the cities, might as well put them to use. I work on graph mining problems. I've done some work with Intel Research Lab on actually developing a new type of processor called Hive that works very differently than the type of processors, the Intel Xeons and i7s that we use today that are specifically designed for working on graphs. I do machine learning work, applied machine learning work on different problems such as kidney health screening and autism prediction. And I've worked on smart city problems, like trying to figure out where should we place the wireless nodes in a citywide wireless network. What is the best placement for those nodes so that you have a reliable network? And more recently, and this is the topic of today's discussion, I started working on computational genomics. I mentioned Iran was actually the one that invited me to this workshop 3 years ago, and then I started going there or coming here, and in this workshop, I learned about this problem. And so I'm a direct, I guess, descendant of this workshop as far as work on this problem is concerned. So as an outline, what we're going to talk about today, I'm going to tell you about open modification search and why it's important, just give you a little bit of background to the problem and why it's difficult and what is the sort of state-of-the-art solution for it. Then I'm going to tell you about how I'm working to improve that, and I'm going to sort of wave and say, here are some anecdotal preliminary results. All right, so what are spectra? Some of you, actually by show of hands, who knows about mass spectrometry here and understands spectra? Perfect, that's awesome. So I'm not going to spend much time going through this. The idea is you have a biological sample that you want to analyze, so you put through a mass spec machine, and what it does is it breaks up that sample through, by bombarding it with electrons into tiny little chunks. And those chunks are ionized, and then through a magnet, the chunks get closer or further away to a reading source, and by a detector, and by detecting how far away from that reading source is, you can tell what is the mass of that chunk. And then a mass spec outputs really a spectrum, which is the relative abundance of these chunks, their masses, and how much of that type of chunk do you have in the biological sample. So when you run a mass spec experiment, you get a whole bunch of these spectra. The part that you don't know are, what are these spectra, where do they come from? So the problem really that we care to solve is, how can we identify what peptides or what proteins these spectra belong to? And there's a bunch of different ways to solve the problem, but one of the ways that works really well is to use massive databases of spectra that have already been identified, and basically match the spectrum you have against one of those spectra in that massive database. And that's exactly the problem that I chose to work on. So you have this basically classical computer science problem called search. I've done a bunch of work in that area, so I figured, why not? There are certain issues, though, here. One, how do you actually represent the spectra? And two, is it appropriate to just do a simple match? So first things first, how do you represent it? Well, you have this intensity mass chart here, and one way to represent it is to simply take the space of the masses and chunk it and bin it, and then account for the intensities within each bin and call that a feature or a dimension in your space. So in this way, you can transform the spectra into Euclidean vectors. Now, if you can represent the spectra as Euclidean vectors, then you have all sorts of classical methods that you can apply to actually do the search. But the question is it appropriate to just apply one of those methods to do a direct search for spectra? And the answer is not so much. Why? Well, because of these, actually you guys know a lot better than me, but a direct match of those vectors won't account for things like post-translational modifications or amino acid mutations and things like that. Instead, some clever people a few years ago came out with this thing called open modification search, and specifically a different type of matching function. So, in matching vectors, you have these proximity measures that you are often familiar with: Euclidean distance, cosine similarity, and so on and so forth. Instead, this new proximity measure is called shifted dot product, that simply accounts for these potential shifts between these ions or these chunks shifting to a different mass. So here in this chart, you have one of these chunks that has a bunch of shifts that could not be matched using a normal, cosine similarity or Euclidean distance measure, but instead it can be matched using the shifted dot product. All right, so that's one issue. The second issue that we have to deal with is the complete massive size of these databases and the fact that they're growing exponentially. We learned about some of this quote-unquote problem we have yesterday as well in other related fields. The data sets you guys are working with keep growing and growing very fast. So basically, either we come up with an all-knowing computer that can solve the problem super fast, or better yet, or more realistically, we come up with better algorithms to solve these problems faster. Now, in the experiments I'm running, we're working with a library that is the massive KB peptide spectral library. It has 4,226,000 spectra, and it's derived from 30 terabytes of human proteomics data. And the queries that we're running against that database, we have a data set that has 30 human draft proteome samples, 2,200 raw files. That accounts for 24 million spectra. And you can tell that's a bunch of data. So, searching that is non-trivial. You can't quite do it on your laptop. All right, so what's the current state of the art? Well, a couple years ago, I was fortunate enough to meet Bill Noble here at CGSI. And even though he didn't present on this particular problem, I got to talk to him about this problem a little bit, and learned that his student, or postdoc, Wout Bitremiu, and him developed this method called ANN Solo, Approximate Nearest Neighbors Spectral Library search, which basically attempts to solve this problem, or is the current state-of-the-art way of solving this problem. The way it works is by using a cascade approach and matching, instead of matching the spectra directly, it matches a vector representation via an approximate nearest neighbor algorithm. Now that poses a couple different challenges. One, by using an approximate algorithm, you may miss some of the spectra that you're actually looking for. And two, by changing from the original spectral representation to Euclidean vectors, you may miss some of those matches again. And they do indeed have those problems. Now, the latest version of that software just came out and uses GPUs to further improve the efficiency of finding the spectra. So we, in my lab, basically reproduced the experiments that they ran on the data set that we just talked about. And for those 24 million queries, the total time was 1.1 million seconds. So about 1 week, 6 days, 15 hours, 1 minute and 45 seconds. That's a whole bunch of time. Luckily, we had a cluster, so it took about a day on 15 GPUs, 15 P100 NVIDIA GPUs. Now, obviously, that's a bunch of time. And my goal is to make that faster. And hopefully also better, find more matches. One of the issues was that, even though we have 24 million queries, only 14 of them were matched to a library spectra. And out of those, 9.7 million were matched using cosine similarity directly, but 4.2 million were matched only by using shifted dot product. Okay. So I've given you an overview of what the problem is and sort of what is the best solution to date. And what I want to tell you now a little bit about is how we can potentially make this better. So how can we improve open modification search? So I mentioned this before. This is a classic nearest neighbor search problem. The idea is you have a point Q, a query, somewhere in Euclidean space, and you want to find within a large set of other points what is the closest point. Or you have some variations of that problem, find the K nearest neighbor points, or find all the neighbors within some distance, or within some similarity threshold from your query point. So there's tons of different methods that have been developed over the last 40, 50 years. And there's tons of methods that work really well for small dimensional data. You guys are probably very familiar with those. Now, for high dimensional data, most people end up using an approximate nearest neighbor method. Things like locality sensitive hashing or ANN, FLAN, so on and so forth. Why? Because it's really time consuming to compute all those dot products, all those things in high dimensions. Now, one of the things that people in general have a hard time with are high dimensional vectors that are sparse. Spectra fall in that category. You have very high dimensionality, but very few non-zeros in general. You don't have a whole lot of non-zero values within each vector. So as such, there's a whole nother class of methods that a lot of people are not very familiar with that I've done a bunch of work on called filtering-based methods that work specifically on sparse data and asymmetric proximity measures. So to understand this, you have to realize that most proximities that we work with are basically some sort of function of the dot product between two vectors. And the way that these methods work are by finding all sorts of theoretical upper bounds on the similarity on these dot products between vectors. So we can use things like the sparsity, the vector lengths, the vector angles, and angles and lengths together, and things like that to throw away a lot of the space and only search a subset of the space. So a lot of the dot products that we would otherwise have to compute, we simply ignore. And by doing that, we can gain a lot of efficiency. So as I mentioned before, here's a couple different proximity measures we might use. The bottom one is the cosine similarity. It's the dot product divided by the product of the norms of the two vectors. The top one is Tanimoto coefficient. It's the dot product divided by the sum of the square norms minus the dot product. And specifically, the dot product is defined as the sum of products. Probably all of you know. But in a sparse vector, think about vectors d2 and d3 over there. In the figure, the dark cells have non-zero values, the white cells have zero values. So you can see that really only the first product matters. Everything else is either zeros or 0 times something, which is still 0, right? So if we can somehow manage to only account for those non-zeros that matter, then we can gain a lot of efficiency. So I'm going to not spend a lot of time on this, but just to give you an idea, there's a data structure called an inverted index that helps you basically take advantage of this non-zero structure in the data. So you can basically build an inverted index and then use another data structure called an accumulator to execute a dot product between a vector and the entire database all at once. And you can do so efficiently by only visiting the non-zeros and ignoring all the zeros. Now, for cosine similarity, one of the things, there's more that we can do. So one of the things we can do is we can leverage similarity estimates. There's this thing called Cauchy-Schwarz inequality that says that the dot product between two vectors is smaller or equal than the product of the two norms. Now, for cosine similarity, we often normalize the vectors, so that doesn't really give us anything, right? It says that, because all the vectors are normalized, the product of the L2 norms is 1. Well, that's the upper bound of the cosine similarity. That doesn't help, does it? But consider these two vectors here, the query and the candidate vectors here. We can, sort of think of this space as being split at some point P, where you have vectors to the left and vectors to the right. I call those prefix and suffix vectors. And guess what? Cauchy-Schwarz inequality applies in that space as well. So we can actually apply Cauchy-Schwarz inequality on the right-hand side and get an estimate of the dot products of that space. We compute the stuff on the left-hand side, and if we add those things together, then we get an upper bound estimate of the similarity. Now, if that upper bound estimate is actually lower than the similarity that we care about, then we can say, hey, these guys will never actually meet my minimum threshold, and we can throw away that pair and move on, right? So a lot of the pruning or filtering that my software does uses these types of techniques. Lots of different type of pruning, as well as data structures that are built specifically to account for this type of pruning. So another idea here is we don't actually have to consider all the non-zeros in the data altogether. So if we have some minimum threshold that we care about, epsilon, then it turns out we don't have to index the entire vector. We only have to index some of the non-zeros towards the end of the vector, as long as the prefix vector norm is actually smaller than that threshold epsilon. By doing this, our inverted index becomes very small, potentially, if epsilon is high enough. And the initial stage of the search is very, very fast. And then we only have to consider very few candidates instead of the entire space. All right. Okay. So in general, the software, which I call here L2 Search, works by creating spectral sparse vectors. We normalize those vectors, then we construct a partial inverted index from that database, and then for each query, we generate a list of candidates from that partial inverted index. We use the remaining part, the unindexed version of those candidates to finish up the computation of the dot products. And then along the way, we do a bunch more pruning using a bunch more of these pruning bounds. So in the end, we have a subset of the dot products that are actually a superset of the true neighbors that we look for, but generally a very close superset. So as such, we can, very efficiently solve this search problem. Now, the current version is basically using the same idea that Wood put forward, which was we use a cosine-based search strategy, find a large set of candidates, and then we apply the shifted dot product proximity on that set of candidates, say 1,000 or 10,000 for each query, and then choose the top neighbor from that second step. The goal, my goal in this project, is actually to develop a filtering strategy that works directly for the shifted dot product proximity measure. So instead of actually searching in cosine space, I can actually search directly for shifted dot products and filter a lot of the space along the way. So that's yet to come. All right, so preliminary results. Actually, TBA. So I was really hoping to show you guys some preliminary results at this meeting. Unfortunately, I realized yesterday that there was some bug in my code and they aren't correct, so I'm not going to show them. But please look forward to a preprint coming out in a few weeks. I also have a tutorial on nearest neighbor search at KDD coming up in a couple weeks, in case you happen to go to Anchorage, Alaska. And then after the preprint, the plan is to release a package that y'all can use through Python and things like that. If you do work in this space and you are interested in this software, please drop me an email and I'll keep you informed of its progress. Instead of the actual results in this project, I'm going to instead give you sort of a quick overview of some of the previous results I've had using cosine similarity instead of not necessarily applied to spectral spectra, just to give you an idea of how well this software can work. So L2AP is the first software that I designed for this problem, and I'm going to show you some results from comparing this against the state-of-the-art at the time, which is a method called join, as well as an approximate method. So, by the way, if I haven't made this clear, which often I forget to do so, these methods I'm working on are actually exact methods. So what you get is the full set of true neighbors. You don't miss out on any neighbors. Versus approximate nearest neighbor methods, you don't necessarily get all the neighbors. You get some of the nearest neighbors, but not necessarily all the nearest neighbors. So we also compared against one of those methods called Bayesian LSH, which was designed specifically for this all-pair search problem. And the data sets that we searched on were either graph data sets, the last three on the bottom, or text-based data sets, the first three, with a fairly large number in the tens to hundreds of millions of non-zeros for each data set. And just to give you an idea of how well this works, I talked about that idea of partial indexing, right? So epsilon here is the minimum similarity threshold that I care about. And index join is the classical method that indexes all the non-zeros. It's sort of the baseline. And then our method, L2AP, this graph shows the number of non-zeros that we index as a percentage of basically the full set of non-zeros for different epsilon thresholds. As you can see, as the threshold increases, the percentage gets very, very small. At 0.95, we index much less than 1% of the non-zeros, which makes for a very small index, which makes for very, very fast searches. And then in addition to that, this shows the type of pruning that we do. So the gray bar represents all the stuff that we did consider, all the candidates that we did consider, but we pruned. And the red stuff are the actual dot products that we fully computed. And as you can see, that is a very, very small percentage. So most of the stuff that we did consider, which again is a subset of all the full set of dot products that we should have considered, is pruned, and most of it is actually pruned fairly quickly in the pruning strategy. So as a result, the software works really well. So L2AP here is in red, the red line, and we get about an order of magnitude faster results than some of the than join at different thresholds. They come close to us towards the epsilon as epsilon tends to 1, but as epsilon tends to 0.5, we get much better results. What's interesting is locality-sensitive hashing, that approximate method that we talked about. For some data sets, like these network data sets like WikiLinks and ORCID links, it does orders of magnitude worse than our filtering-based methods. And why is that? Well, because those data sets have really, really high dimensionality. So the only way that really those methods can work well is if you have a fairly small dimensionality. So it might have worked better if I applied some sort of dimensionality reduction first and then applied LSH. But in this case, we applied LSH directly on the sparse vectors. And I have a parallel version of L2AP, and these graphs show that it scales very well linearly as the number of threads increases. And then A few more results from a different version of the software that actually deals with a different proximity measure called the Tanimoto coefficient. So here we deal with chemical compounds. So we have a bunch of data sets that some of you might be more familiar with, like MLSMR and SC, but also some text data sets like patents and RCV1. Some of these data sets are really large. So 11.5 million compounds for SC with 1.7 billion non-zeros. And a bunch of different subsets of that. So again, we compared against a bunch of baselines. And again, this shows you the type of pruning that we do. As the threshold gets closer to 0.99, let's see, yeah. Then most of the stuff gets pruned in the first stage. L2CG is the first stage of pruning that we do. PS is a pruning bound that we check right after that first stage only once. And VLA is a length-based filter that, again, we apply right after PS. And L2CV are checked in the verification stage. And most of everything is pruned, and the amount of stuff that we actually compute is very low. Now, for these chemical-based data sets, you have some very interesting differences between chemical-based data sets and the text-based data sets. For text-based data sets, the amount of true neighbors is actually very, very small. But for the chemical-based data sets, the amount of true neighbors becomes quite large for the lower similarities. So the similarity threshold is not this, random thing that you can just set, 0.9 for everything. No, for different data sets, you actually have different thresholds that are meaningful. So that's something to keep in mind. Okay, and again, our software here is called TAPNN, it's in red, and we get performances an order of magnitude, up to an order of magnitude faster than previous thresholds. All right, so, and this shows that unlike the previous software, our software can scale to these very large data sets like SC. Here I only show, for very high thresholds, and yeah, it did take about a day or a couple days for the full data set. Now, here's the tricky part. That's using only one thread. So if I, make this a parallel algorithm, then or run it on a cluster, then I can search those very large data sets much more quickly. All right. So I hope I've given you a quick overview of. I've succeeded in giving you a quick overview of open modification search. And if you have any questions, feel free to ask.